We went through the stages of mitosis. What are the stages of mitosis? Prophase, metaphase, anaphase, telophase. Okay, that's all well and good. Well, what are the characteristics of each of those phases? Prophase, characterized by what? Condensing what? Okay, chromosomes condense from what? Chromatid. And what is chromatid? DNA with proteins. So are chromosomes DNA and protein too? Yes or no? Yes. All right, so it's really a mixture of DNA, proteins condensed together. All right, so prophase, chromosomes condense. What's next? Metaphase, characterized by what? How do you know a cell's in metaphase? Right, so the chromosomes line up along the equator of a cell, forming the metaphase plate. How do the chromosomes line up along the equator? How do they get there? Microtubules push and pull them so that they're aligned, right? How do microtubules connect to the chromosomes? At the centromere, through what? No, not through centrosomes. Asters are at the poles, right? Where those are where the microtubules are coming from. Okay, microtubules connect to the chromosomes at the centromere. Where's the centromere? Right, so we have our chromosomes that look like this, right? And these are called what? Sister chromatids. Now our microtubules connect to the centromere, right? How do they actually connect to the centromere? There's a special name for the connection. And we went over it. Kinetochores. Okay? So that's the link between microtubules and the centromere. All right, so we put a little arrow there called the kinetochore. Way to remember that. What's going to happen to the chromosomes after connection? After they line up in the metaphase plate, what's going to happen to the chromosomes? They're going to be pulled apart. Okay? So, is that a movement? Yes? Right? That's a motion. What type of energy is related to motion? Kinetic energy. That's why it's called a kinetochore. Right? It's attached. There's going to be some movement that's attached to that. Okay? So that's one way to remember that. So the kinetochore is attached to microtubules to the centromere. Once there's a separation between these sister chromatids, we've moved from metaphase to anaphase. All right, so chromosomes separate. Okay, they start migrating towards the poles. That's anaphase. When they reach the poles, what do we have next? Well, so anaphase begins when, as you get migration or separation of the sister chromatids. Telophase begins when the chromosomes reach the poles, the ends, and then they begin to decondense back into chromatids. Right? So we go into telophase, and what happens? We end up in cytokinesis. And cytokinesis is technically what? Separation of the cytoplasm. So we really have two processes that are ongoing. Mitosis, right, which is a nuclear division. Okay, nuclear. And then we have I still have not mastered the writing on the iPad, but which is a separation of the cytoplasm. All right. Nuclear division or separation of the chromosomes is directly related to what part of the cytoskeleton? Division of the chromosomes, separation of chromosomes, is directly related to what part of the cytoskeleton? What filaments are responsible for that? Microtubules. Okay. All right, and microtubules are composed of what type of protein? Tubulin. Okay. Is it understandable that another part of a separate part of the cytoskeleton would be related to or would control cytokinesis? Is that kind of logical? All right, and that's exactly what happens. So you have microtubules, one type of protein that's going to control separation of the chromosomes, and you have another type of protein filament that's going to control separation of the cytoplasm. All right, so let's do cytokinesis. All right, so here's cytokinesis. This is, we have two nuclei that have separated, and they're now beginning to form. What's happening here? What's happening at this point? What has reformed? The nuclear envelope. All right, and what is this? Cleavage furrow. Okay, in a way, we call it the cleavage furrow because it's going to separate into two cells. And cleavage is also another name for rapid mitotic divisions. Okay, so as it happens very quickly, it's separation in between. All right, so this is called the cleavage furrow. It's the beginning, right, the infold of the separation of the cytoplasm. Okay, how this occurs is that it's sort of like tying a shoelace, tying a loop in a string, and pulling the loop tight. Right, it contracts around around a ring. Right? This is a contractile ring. All right, so you're going to get, let's erase this. All right, so you get indentation not on a two-dimensional plane, but on a three-dimensional plane. All right, so it's going to indent here, 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 and also on the back side. Right, so every sort of a ring is going to contract. All right, so it's going to get smaller and smaller and smaller until eventually the phospholipids form two separate membranes. Okay, so what is occurring around this contractile ring is that actin microfilaments are actually pulling the plasma membrane in closer as you get contraction of the ring. Okay, and actin microfilaments are a separate type of cytoskeleton filament, right? So one cytoskeleton filament works on separating the chromosomes. Once that occurs, then actin microfilaments work on separating the cytoplasm. Lo and behold, what do you end up with? Two new cells. Okay, so that is mitosis and cytokinesis of eukaryotic cells. Mitosis is a nuclear division. Cytokinesis is a cytoplasmic division. So let me ask you a question. What happens if a cell doesn't have a nucleus? Because what we've done is we've condensed the DNA and the protein, everything that's found inside the nucleus, and separate it into two nuclei. But not all cells have nuclei, correct? So what happens if a cell doesn't have a nucleus? How do they divide? Would the process be more or less simple? Would it be more simple or more complex? Let's ask that question. It'd be more simple. Why? There's not as much stuff. What is it? What has to be done in order to separate? Well, let's say separate the DNA, genetic material, of a cell that doesn't have a nucleus. Does it have to break down in a nuclear envelope? No. Does it have to condense into chromosomes? No. All right, so it's a simplified mitosis. Okay, and that's really what we call. All right, first off, here's some pictures from your book about the cleavage furrow. And this is sort of down here trying to show you your contractile ring. This is a scanning electron micrograph of the cleavage furrow. You can see the invagination that occurs at several different points. It kind of gets pulled in. Different from plant cells, so we have eukaryote animal cells versus plant cells, formation of a cellular plate. So something we went over in lab. All right, formation of a cellular plate, right? Reformation of the cellular wall, which will actually physically separate the two cells. All right, so a cleavage furrow doesn't necessarily happen in plants. In prokaryotes now, cells that do not have nuclei, they still need to replicate their DNA, correct? All right, we don't want to produce cells that don't have a full copy of the genetic material. All right? So what you see here is a diagram of DNA replication and separation of the cells. All right? It is a lot less complex. Okay? So here, E. coli, sort of your typical prokaryotic cell. There is a point of DNA replication where it starts. Okay, so that's what it's calling the origin of replication. And maybe you can see this better if I shut the light on. I don't want you to fall asleep though. Okay, so the origin of replication you see up there in green. In bacterial chromosomes or prokaryotic chromosomes, for the most part, there's one circular piece of DNA. It's not like eukaryotic DNA where there's chromosome 1, X chromosome over here, Y chromosome over there, right? In eukaryotic DNA, specifically humans, what we're talking about, each chromosome in eukaryotic DNA has its own start site of where DNA replication is going to begin. Okay? In more simplified chromosomes, genetic material such as bacteria, there's one spot. Okay? So there's an origin of replication. It's going to proceed in both directions from that origin, eventually forming another circular chromosome. Okay? So you get two copies of the origin. This is where DNA replication, let's do this. DNA replication is proceeding, and as then you get replication finishes, cells split. Okay? Splitting of the cells in this case is dependent upon the structure of the membrane and whether or not, and the cell wall that prokaryotes have. Okay? But that is a lesson in the specifics you can learn in microbiology. Okay? Eventually, what you're saying is there's no nuclear envelope breakdown, no point in condensing DNA, there's no point in separating chromosomes or having microtubules that will pull them apart because we just have one circular piece of DNA. Okay? There's no proteins that are condensing this DNA either. Okay? So it's in a way, simplified mitosis. This is binary fission. All right? Binary, splitting means two. Fission means to split. All right? So binary fission, splitting into two. Going to replicate the DNA, split, and produce two cells. Questions on binary fission? So what do you think happened first? Evolutionarily, right? In the sense of evolution, what came first? Binary fission or mitosis? Binary fission. We know that prokaryotes evolved first and eukaryotes evolved from prokaryotes. So as the genomes got more complex, more genes, more DNA, more chromosomes, protein that's involved with that, the process of mitosis evolved with it. Okay? Questions on mitosis and binary fission? All right, so the other day, we also went over the cell cycle. Right? We introduced the cell cycle and mitosis as a very small part of the cell cycle. Right? And I said, what was the cell cycle? So we had, let's say we have mitosis. Right? We'll call this the M phase of the cell cycle. After mitosis, the cell then goes into what part of the cell cycle? G1. G1, G stands for gap phase, growth phase. Okay? After G phase, it goes into, all right, the S phase. And S stands for synthesis. Synthesis of what? DNA. All right, so this is where DNA is going to replicate. After S phase goes into G2. G2 standing for gap or growth phase 2. Okay? And after G2, cells then go into the nuclear division, mitosis, and including in mitosis will include cytokinesis as part of that. Okay? This is the cell cycle. All right? What do you think are the signals that tell the cell to go through the cell cycle? Where do you think they come from?
Outside the cell.
Think outside the cell. Okay. So let's do this. So if I were to give you this data, okay? So this is an experiment taken from your book. All right? Experiment 1. And for whatever reason, however, you don't have to get into specifics of how this occurred. Okay? Researchers could take a cell nucleus. All right? A nucleus from a cell that was in S phase. Okay? And a cell that was in G1. What's the difference of what's happening in those cells? What's happening in S phase? DNA is being synthesized. Okay? What's happening in G1? It's just kind of growing. Okay? So if I took this nucleus from S or fuse these cells together, all right, what happens is that this G1 cell immediately goes into S phase, regardless of what time I took it from. Okay? So all right, so that kind of gives you an idea of what's happening. Let's look at the other experiment 2. Take a nucleus from a cell that's undergoing mitosis, and I fuse it with another cell that's undergoing G1. Okay? And immediately, that G1 nucleus starts to undergo mitosis. What conclusions can you make from this? What's happening in this nucleus? What's happening in these cells? These nuclei are inherently different, right? The G1, the cell that's in G1, does not have the machinery set up to undergo DNA replication or mitosis, right? It's not ready yet. But when you fuse the cells, you mix the cytoplasm, what occurs? That G1 cell is stimulated to behave like the other nucleus. What does that tell you? Alex, you have a question? All right, so it's the stage that's further ahead. Okay? Jordan. All right, so if that were the case, what do you think would happen if I fuse 2 G1 cells together? Would it immediately go to mitosis? No, it'd stay in G1, right? There's G1, fuse the 2 together. All right, so there has to be something that's present in these cells, right, in the S phase cell or in the M phase cell, that is stimulating the G1 cell to begin division. Right? Signals from the cytoplasm. Why? Why the cytoplasm? Because it's shared between both cells, and everything that's inside the G1 cell is composed or held inside a nuclear envelope. Same thing here with the S phase cell, right? So it's not something that's necessary in the nucleus because those nuclei are still separate. Okay? But in the M phase cells, those nuclear envelopes broken down so stuff can be kind of everywhere. So molecules present in the cytoplasm actually drive progression of the cell cycle. All right? this is just a point to show that molecules outside of the machinery within the nucleus are going to drive the cell cycle. All right? And there are inputs, okay, that tell the cell to go through G1, begin to divide, right, begin to synthesize DNA, go through G2. There's checkpoints that control the progression of the cell through the cell cycle. Okay? Questions on that? So here is an outline of, well, a diagram that's in your book about the cell cycle itself. This is the control system. Okay? You see those red little gates up there. Those are checkpoints. All right? And I kind of mentioned this on Monday about this being the quality control system for the cell. All right? The cell will go through its normal life cycle as long as the environment is conducive to it and there's enough input and progression to push them through these restriction points. Or I shouldn't say restriction points, I should say checkpoints, right? Because there's one specific checkpoint that is the restriction point. Okay? So you have quality control points. G1, all right? At the end of G1, we're going for transition from G1 into S phase. What do you think the cell needs to go from G1 to S phase? A certain size, okay? What else? Think about what's about to happen in S phase. What's going to occur in S phase? Synthesis of DNA. So in order for the cell to begin to make DNA or replicate DNA and progress through this checkpoint, does it need all the machinery necessary to synthesize DNA? Yes. All right. It also takes in, so it needs all the internal stuff, right, to engineer DNA, and it also needs these external signals, growth factors, the environmental signals, etc., to push it through that checkpoint. Okay? Once it passes, this is called the restriction point, right, G1. Once it passes G1, it is destined to replicate DNA. All right, so once it goes into S phase, it's going to replicate all its DNA. It's going to go through S, it's going to go through G2, and you get to this G2 checkpoint. What do you think it's checking there? What's happened in S phase? DNA synthesized. In G2, we get more growth. So what is happening at the G2? What is it checking for at the G2 checkpoint? How well, so completeness or quality control of the DNA replication, are the necessary things in place for mitosis to occur. All right, we're going to check all of that. If it fails a checkpoint, what do you think happens? Does it just, okay, well, what would quality control if you're working at a factory and a computer doesn't pass quality control? What happens to it? Do you send it? Do you still sell it off to the market? Your iPhone, does it get sent to you? No, it gets what? It gets trashed. Okay, or broken down and replaced. It gets recycled in some way. That's what happens with the cells. If it doesn't fail, if it fails a checkpoint, it undergoes apoptosis, right? Which is programmed cell death. Don't want to pass on incomplete or incomplete information, sub-quality cells onto the next generation. Okay? So we have G2 checkpoint, and there's also an M checkpoint in mitosis. Right? So during mitosis, when you get separation of chromosomes, all right, if there's not attachment of microtubules to chromosomes, the cell won't divide. If there's incomplete attachment, if the microtubules don't all attach, then they won't divide. Will stop cells in mitosis. All right, so there's a quality control points as this goes through. Okay? Now, this is a complicated point. Let's move that out of the way. This is a diagram that shows how complicated this is and what proteins are involved in this. All right, so the cycle's a little off. You see G1, S phase, G2, and M. Okay? What you see is there are factors that are positively regulating, right, showing progression of the cell cycle, making the cell move towards mitosis, and then you have inhibitors of that, or negative regulators. You have these other on the inside of the circle showing sort of brakes. Okay? So you have positive regulators that are like the gas pedal, pushing the cell cycle forward. Okay? And you have negative regulators that are like the brake and pushing and slowing it down. Okay? These are all what we call positive regulators of the cell cycle are cyclins. You see cyclin D, okay, cyclin E, cyclin A, cyclin B. These are small proteins that will be altered. Okay, they come together with their partners. You see those what are called CDKs. All right, so you see these CDKs right here. These are kinases. A kinase is an enzyme that will phosphorylate. What do you think phosphorylate means? Addition of a phosphate group. So a kinase is an enzyme that adds a phosphate group to something. Okay? So when you add a phosphate group, this cyclin D and CDK4, they complex together and make an active kinase. All right? This complex, okay, will phosphorylate this protein right here called retinoblastoma. Right? And that will push the cell cycle through G1. That's one mechanism of moving past the restriction point and the G1S transition. Okay? These others, these are inhibitor kinases on the inside, P27, P21. Those are what we call tumor, and RB are tumor suppressors. Why are they called tumor suppressors? Why aren't they called negative regulators?
Because if they let the cell pass, then tumors occur.
Right, so that's how they were originally found. They were found in cancer tumors, right? And a lot of times you have mutations in these genes that allow the cell to go through this restriction point faster and faster and faster. Right? You remove the brake, the car goes down the hill pretty fast, right? Same thing that occurs. Okay? You remove the brake, the cell goes through the cell cycle faster, and that's what you end up with cancer. All right, so retinoblastoma is RB, that protein right there, is commonly found in childhood cancer of the eye, right, where the mutation is found in the eye. That's why it's called retinoblastoma. Other ones that you may have heard of, what's other stuff? P21, P27, you may have heard of, you may see in literature. P53 is a big one. You may have heard of BRCA1, BRAC1 and 2. That's breast cancer related gene. Right? Those are all tumor suppressors that work to regulate this progression. Okay? Questions on cell cycle progression and inhibition?